Well, good morning. Welcome to the video on how to sample oaks for sudden oak death. My name is Matteo Garbellotto, and I am a professor at UC Berkeley. And today we're going to go through all the things that you need to know about sampling oaks for sudden oak death, commonly known as SOD. So the first thing I would like to start from is all the things that you will need in order to properly sample oaks. And the first thing you will need is something that you will have received upon request from us at Berkeley, from the Forest Pathology Lab. And it is Petri dishes with a specific growth medium that's used to grow the sudden oak death pathogen. In a package, you will receive 20 such plates and a form that comes with the package where each specific Petri is labeled with a specific code, which you will need to use during the processing of the sampling procedure. So these are the first two things you will need. And then you will need to bring a few things of your own. And these are a tool to take off the bark from the oak. In this case, our preference is to use a sharp hatchet. A knife, sharp again, to sample wood under the bark, and we'll look at that wood later on. A pair of tweezers that you may need to select a little chip of wood to put on the Petri dish. A flashlight that may help you to identify where to cut the bark, especially when it's dark. A Sharpie that you will need to label the Petri dish that you're using, which is the Petri dish where you're going to place the little chip of wood. A pencil to fill in the rest of the form, and we'll talk about that a little bit later. Some ethanol or rubbing alcohol, 75% possibly, that you will need to sterilize the tools, and some tape to make sure the lid doesn't come off from the Petri dish after you've placed the chip of wood inside the Petri dish. You may also want to have a cell phone, which you will use in two different ways. First of all, you will use it to look at the video that we're shooting right now, just to be sure that you're following the right procedure. And secondly, you can use the phone with the app SOD Map Mobile to make sure that you're sampling a tree in an area that's reported to have sudden oak death. Of course, you may be sampling outside of an area with sudden oak death, but 99.9% of trees that have the disease are within areas that are known to already have an infestation by this particular disease. And of course, you may want to have gloves when you use sharp tools so that you don't injure yourself in the process. Now we're going to talk a little bit about site and tree selection for sampling. The first thing you need to do, as I mentioned earlier on, is make sure that you're in an area that it's known to be infested by sudden oak death, and the best way to do it is to use the app on your phone that's called SOD Map Mobile. And by using the application, for instance, in this case, I'm using the application, and by tapping the risk button, I can see that I am in an area at medium risk for sudden oak death, which means that the infestation is known to be present in the area. So that's a good start. The second thing, you probably want to work on a day like this when it's not raining, because rain may actually mask the symptoms on the bark, and so it may be really difficult for you to identify where to peel that bark off so that you can get to the wood under the bark. And then third thing, in 90% of cases, you will see, or there should be, a California bay laurel next to the oak that you're sampling. And California bay laurel should have symptoms like the symptom that I'm showing here. I hope you can see it. So these are the symptoms that you should see on a California bay laurel next to the oak. However, the symptoms on the bay laurel may be gone by the time you sample the oak. So the most important thing is simply to know that there is a bay laurel next to the oaks that you're sampling. Here we are in Berkeley on a day when it's not raining yet. We have two oaks behind us, and there's plenty of bay laurel around the area. SOD map mobile tells me the risk for sudden death is medium in this specific location, so we know the infestation is present in this area. But only one of the two trees behind me is a tree that we should be sampling. And I will tell you why in a second. Here we are next to two oak trees that are not doing very well. You cannot see it from that point of view, but the crowns are fairly poor, and we suspect these trees have been infected by the sudden death pathogen for at least 1 or maybe even 2 years. However, the tree on my right-hand side is already dead. Basically, most of the foliage is dry, and there are several fruit bodies of a fungus known as Annulohypoxylon thorsianum that are growing on the stem. And they look like charcoal balls, charcoal spheres that are growing out of the bark. And when these charcoal spheres grow out of the bark on the main stem of the tree, that is a very good indication that the tree is already dead. The same applies to trees that have sawdust, on the bark. That sawdust is not produced by a fungus. The sawdust is produced by tunneling bark beetles, and that's also another indication that the tree is already dead. That is not a tree that will yield any positive results for sudden death. In order for a tree to be a good tree to be sampled, the tree still needs to be alive. Luckily, the tree on my left-hand side here, with a thinning crown, is still alive. It has green leaves. It doesn't have Hypoxylon thorsianum growing out of the main stem, and it has very typical diagnostic bleeding lesions on the main stem that we normally associate with sudden death infection or with infection by other Phytophthora species. Let me show you what these lesions look like. Okay, here we are on the other side of the tree. It's actually the side that's closest to the bay laurel that probably infected this oak. And I'm using a flashlight to actually highlight the presence of a dark lesion on the outer part of the bark. This is dried out sap. And this sap that you see here, so you see the brown darker area, this was covered by sap that was produced by the tree because of the infection process that's undergoing under the bark. So here you go. Hopefully you can see that we have a very distinct brown lesion. It's mostly dried, but if you look carefully, you should be able to see some little drops of sap still coming out. I'm highlighting one right here. So that's a little drop, and that's a drop of sap that's still coming out from the tree that's infected. Somewhere under this brown lesion, we need to identify the margin of the infection by the sudden death pathogen. So somewhere under this brown lesion, we need to identify where the margin of the infection by the sudden death pathogen is, because that's the only area that we can sample to positively obtain confirmation that this indeed is a tree infected by sudden death. So the brown area on top of the bark gives us an indication of where the lesion may be under the bark, but it's just an approximate indication. So we actually need to carefully debark the tree at the edges of the brown lesion outside until we can see the edges of the lesion under the bark itself. And that's where we're going to perform our sampling. Okay, so now that we have identified the possible area where the lesion may still be growing under the bark, we actually need to debark the tree partially to expose the area that's infected by the pathogen under it. So I'm going to try to do, oh my God, look at this. This is extremely clear here. Okay, so here you have a perfect example of a margin of a lesion that's undergoing, and it's basically girdling the tree. And on the right-hand side, you have a darker area that's currently colonized by the sudden death pathogen. And on the left-hand side, you have still healthy wood and healthy cambium under the bark. In between, you can clearly identify a line. The line is dark, and it's not really a straight line. So if you can take a closer look, you can see that the line has little curves. It's irregular. There are little curves, little bumps, and so forth. This dark line, it's the place where you want to collect the wood that you will place in the petri dishes that you obtain from UC Berkeley. It's important to be sure that the tools we use to debark and then sample the wood are clean. So here I'm showing you how you can quickly achieve that goal by simply having some alcohol. I put some alcohol here in the spray bottle. I spray the blade of the knife. I did the same thing with the hatchet before. And then I can simply dry it with a clean paper towel. I dry the blade on the knife. And at this point, I am ready to sample the lesion. Okay, so the most important thing is to select the portion of the oak wood that's in between the healthy area on the left and the unhealthy area on the right. So basically, you want to cut a chip, there you go, that is right in between the two. And you can use your knife to cut a few chips. You will need 4 per tree. So if you're able to do all 4 at once, here we go. We have pre-prepared 4 chips to be plated by using the knife on the part of the stem that we have exposed. Okay, here I am, and I will pull off one of these chips at a time using tweezers that I've previously cleaned using the alcohol. Here it is. I picked one chip, and now I'm going to show you how to put this chip inside the growth medium that's in the petri dishes you received from Berkeley. At this point, I need to place the chip that I collected from the oak into the petri dish. So the petri dish will still be closed at this point. When I'm ready, so when I have the chip in the tweezers, I open the petri dish, I place the chip on the surface, and then gently I submerge the chip inside the medium, okay? So that it's mostly inside the gel. It's a kind of a gel, and you want to put the chip as much as possible, not on top of the gel, but inside the gel. Now, once you're done, you will close the petri dish, and then you will place 3 more chips from the same tree at the corners of an imaginary square that fits within the petri dishes. And I'll show you what a fully utilized petri dish should use. Again, you should place 4 chips from any single oak that you sample on the same petri dish. This is a plate that's being fully utilized to sample the tree. Again, imagine a square inside the round petri dish. At the corners of the square, you will place 1 chip. The chip goes under or inside the gel medium. And then at this point, you close firmly the lid on the petri dish. So at this point, the lid is firmly closed. Then you turn the plate gently upside down, and then you look at the list that you received from Berkeley. If this is the first petri dish that you use, and then you look at the first row on your sheet, and in this case, the first row says A0001. That is the code that goes with this particular petri dish. At this point, your sampling is completed. You will still have to fill in data right after you finish writing the label on your petri dish. But before you do that, we ask you that you cut 2 small pieces of masking tape, and you place them in between the lid and the back of the petri dish so that the petri dish will not accidentally become open. Okay, at this point, the samples, the 4 chips of wood from the oak, the same oak, are on 1 petri dish. The petri dish has been labeled following the instructions on the sheet that you received from Berkeley. The plate has been sealed. We're using 2 pieces of masking tape. So at this point, I need to fill in the information about this particular tree and this particular sample. So I go to the sheet that you received from Berkeley on the first row because it's the first plate A0001, again, plate A0001, And the information that's needed includes the latitude and the longitude, so the GPS coordinates, the tree species, and the diameter of the stem that I sampled, and finally the date of sampling. So this is all the information that I need to record. Once I'm done with recording this information, here I am writing down all this information that I previously obtained. Then I'm ready to go on and sample the next tree. We actually have sampled 18 stems in this area. So I have 18 petri dishes with wood chips collected the way we have just shown you. I put all of the petri dishes that I used inside the sleeve that I received from UC Berkeley. And I need to keep the plates in a cool and dry place, so it could be a cooler with some blue ice. It could also be a fridge. And these samples have to be shipped to the address in the form you received from UC Berkeley within 48 hours from collection. So again, they have to be shipped using next day service within 48 hours from collection. And the day the samples are shipped, it is very strongly advised to email information about the shipment to the contact person that's also listed on the form that you received from UC Berkeley. The petri dishes you sent to UC Berkeley will be processed, and you will have results within 6 weeks from receival of the petri dishes. My name is Matteo Garbalato. I'm a professor at UC Berkeley, and I thank you for participating in the SOD oak testing program.